Good evening, ma'am.
Good evening, children. So yesterday we have completed till the nucleus and the cytoplasm, also cell wall. Cell wall, nucleus, and cytoplasm we have discussed. Now we'll be going to the next one. Just a minute, children, just a minute. Just give me 2 to 3 minutes, okay? Okay, so next topic we will be discussing now. Now the next topic is the endoplasmic reticulum. Let me share the screen. Is my screen visible?
Yes, ma'am.
Okay. The first one, what we are going to discuss is the endoplasmic reticulum, which is also known as ER, endoplasmic reticulum. It's called as the ER. Now, when we talk about this ER, it is basically the network of the membrane, which will be present in the cytoplasm. So, in the cytoplasm, this endoplasmic reticulum will be present all around the cytoplasm. It is the network of membranes present in the cytoplasm. So in the cytoplasm, there will be the network of membrane that is called as the endoplasmic reticulum. Now, it was discovered, ER was discovered by Porter, Portr, Porter, Claude, and Coulomb. So these were the three people who discovered it. Then these are present in all cells except the prokaryotic cells. These are present in all cells except prokaryotes. So it has three components are there. Three components are there for the ER, which are basically the cisternae, then vesicle, and then the last one are the tubules. Cisternae, vesicles, and tubules. Though these are the three components. Cisternae are like this structures, like this. These structures will be there, which have the ribosomes over them. Ribosomes, these are what? These are the cisternae. The second one, the vesicles will be somewhere oval or the spherical structures. They also have what? They also have the ribosomes. And the last one, as the name says, they are the tubules. They will be in the form of the tubes-like structures, like this, or it can be like this also. So they are the tubules. So cisternae, they are basically the long, flat, and unbranched tubules. Whereas the tubules, they can be unbranched, they can be branched as well. And vesicles, they are basically the round or the spherical cells. Now these ER are divided into two types, which are the smooth ER, the smooth endoplasmic reticulum, and the rough ER, rough endoplasmic reticulum. In the smooth one, they are made up of the tubules. So they will have what? Out of the three components, they will have the tubules mainly. And what do? They will be helps in
This is a proteins.
steroid.
Lipids.
Lipids. So helps in steroid lipids formation. And in the smooth, the ribosomes will be absent. So in the case of them, the ribosomes, spelling is wrong. Ribosomes are absent. And also, they helps in membrane biogenesis. So what is membrane biogenesis and what is the function of it? Any
Synthesis.
idea? Come again?
The process by which the membrane is made, membrane synthesis.
Formation of the membrane will take place. Correct. So this is important for the exam. What is membrane biogenesis? So membrane biogenesis is basically the process by which new cell membrane, by which new cell membranes are formed. New cell membranes are formed. And at the same time, whatever the previous membranes are there, they will also be existing there. And existing membranes are maintained in the cell. So those are called, that process is called as the membrane biogenesis. Or in the simple word, we can say that it is how a cell makes and renew its membrane. So they are just developing the new membrane over them. Now, why the formation is needed? Why this membrane biogenesis is needed? It is needed because it helps in the formation of the membrane for the mitochondria. Like mitochondria is there, they also need a membrane.
Double
Nucleus is there,
bond.
they need it. Correct. Nucleus is there, they need a membrane, right? For them, they are very, very helpful to form those kind of the double membrane layer structure. So for first thing is the formation of the new membrane will be done. And at the same time, they also help in the cell growth. First function, formation of new membrane will be there. Second thing is the cell growth. So as a cell will grow, the new membrane material will be required. So this new membrane material, which is through the membrane biogenesis process, they will be giving them that material. Now then, it is also important for the cell division. For cell division, also membrane biogenesis is important. So during the cell division, what happens? The new membranes will be formed for the offspring. Then they are also important to form the new membrane so that the repair and replacement can take place, because every time the cell will not be in the good condition. So there should be some kind of the replacement should be there. So for that reason, the membrane biogenesis plays a very, very important role. Then second one is the rough endoplasmic reticulum. In the case of that, it is made up of cisternae and the vesicle. Made of cisternae and vesicles. So they help
Ribosome,
in,
and that's why it looks rough under the microscope. And because the ribosomes help in protein synthesis, that's why it also helps in membrane biogenesis.
correct. So they actually help in the formation of the protein synthesis, and also they contain what? They contain the ribosomes on their surface. Contains ribosomes on its surface. So because of this, it also helps in the synthesis of protein and also the membrane biogenesis. Then the next one, functions of ER. Keep in mind, children, these two, rough and the smooth. Rough is for the formation of the lipids, whereas the smooth is, sorry, the rough is for the formation of the protein and the smooth one is for the formation of the lipids. So learn the function of it. And function of ER, it is the only organelle which can move within the cell. So whatever the cell is there, it is the only organelle which can move, which can move within a cell. So within the cell, they can be moved. So it serves as a channel. So if they have to transport something, they can send it in the within the cell itself. So that is why it act as a channel for the transport of material transport of materials between various regions of cytoplasm. The second thing they will be doing, they will be helping in the formation of the fats, which is basically the lipids, then steroids, then the protein, synthesis of fats, steroids or cholesterol, etc. will be generated, will be produced because of them. Then they also help SCR, smooth endoplasmic reticulum. They also plays a crucial or important role in the detoxification. This is very important for exam. Which kind of the endoplasmic reticulum help in the detoxification of the drugs? So that is the SCR, detoxification of drugs and poisonous byproduct, because in our body some or the other kind of the toxins will be generated due to the different kind of the eating things and all, so they will be helping in detoxification of that. Then the next one is the Golgi apparatus. Golgi apparatus. So Golgi apparatus, they basically will be like this. They will be somehow like this, kind of these structures. So Golgi apparatus is the system, or it consists of a system. It consists of a system of membrane bound vesicle. Membrane bound vesicles. They will be arranged parallel to each other. So you can see in the diagram also, whatever the stacks are there, they are arranged parallel to each other, and they are called as the cisternae. So these all are called as the cisternae. And also along with the cisternae, there will be some large and spherical vacuole also present around the cisternae. Spherical vacuole. So this one is what? These are the vacuoles, and these are the cisternaes which are present in them. Then it was discovered by Camillo Golgi. It is a single membrane structure. So if they ask you in the exam, name a single membrane organelle, then the answer should be Golgi apparatus. Then it is absent in the prokaryotes. So prokaryotes does not have them. They are absent in prokaryotes. Then the functions of them, they help in the formation of lipids. Formation of lipids, they will be helping. Then they also help in the melanin secretion, formation of
Packaging and like
yes.
stuff of like
Huh.
stuff.
So they also help in melanin secretion or production of it. At the same time, whatever the we can say the modification is required, or the packaging is required, and the storage is required of what? Of the lipids and proteins. So lipids and proteins will be formed by the ER. So those changes which are required, or which are need to be packed, which are need to be stored, that will be done by the Camillo Golgi. So that is the place. Camillo Golgi is the place where the modification of lipids and proteins, packaging of lipids and protein, and storage of lipids and proteins will be done. So it's a kind of the assembly. It's a kind, or we can say it is the site for assembly of new membrane material. So here the new membrane material will be assembled. Now children, just give me somewhere 2 to 3 minutes, okay? Just give me 2 to 3 minutes. Okay. Now the next one is the mitochondria. Mm, Mitochondria. Mitochondria is known as the powerhouse of the cell, and it is a double membrane structure. They are absent in prokaryotes. Double membrane structure. They are the powerhouse of the cell. Or they are also called as storage battery because they help in giving the energy. Then they are found in the cytoplasm, scattering all around cytoplasm. The it was first seen, mitochondria was first seen by Kolliker in insect cells. In the cells of the insects, they were like first identified. And the maximum amount of the mitochondria we can see during the active cells. So when the cells get activated, then the maximum mitochondria will be seen so as they can produce or they can make the lot of the energy in our body. Maximum mitochondria are found in metabolically active cells. Now, they have the structure which will be like this. Inner membrane and outer membrane will be there. This is the outer membrane. Outer membrane. They have the specific kind of the proteins present in them. And then the inner membrane, it is folded inside, which is called as the cristae. And these cristae will help in the respiratory cycle, or we can say the Krebs cycle. And between them, between the outer and the inner membrane, there will be the space which is called as perimitochondrial space. So this is inner membrane, and space between them is called as perimitochondrial space. And the fluid which is present here, the fluid name is called as the matrix. So outer membrane, inner membrane, the space is perimitochondrial space, and the fluid is called as the matrix. Then the functions of the mitochondria. Main function is to produce and store the energy. Whatever the energy is there, they will be storing it in the form of the ATP, and this ATP will be formed by ADP, adenosine diphosphate, plus inorganic phosphate. It will be giving the ATP. Then it is a site of the respiration. So the mitochondria is a place where the respiration will take place. So that is about the mitochondria part of it.